All right, welcome back for more biotechnology. So we have one last topic to discuss in Chapter 4 on DNA, and that is the use of gel electrophoresis to visualize, separate, and study DNA molecules. So we've already established that we can kind of classify DNA according to its size and the number of base pairs. So DNA molecules come in a wide variety of different sizes. So there will come many times that we commonly need to separate DNA molecules from each other according to their size in order to isolate them and deal with what we want to deal with. So in order to do this, we use a technique called gel electrophoresis. So this uses an electrical current that is passed through a gel that contains the DNA molecules, and this causes the DNA molecules to separate based on their electrical charge. Now, the nice thing about this is that all DNA molecules are negatively charged because of the phosphate groups in their backbone. So they're just naturally negatively charged. So because DNA molecules will be proportionally negatively charged according to how long the DNA molecule is and how much of a backbone it's got, what ends up happening is that the DNA molecules really just migrate based on their size. They separate and migrate based on just how many base pairs they contain. So the gel for DNA electrophoresis is made out of a molten agarose solution. So agarose is a type of sugar that comes from seaweed, the same way agar, which we talked about before, it's not quite the same thing. Agar was a protein that comes from seaweed. So the gel is cast out of a molten agarose solution in a rectangular casting tray. So you can kind of see what this looks like right here. And then a comb, which you can kind of see here, that has these little well indentations in it. We cast it into the gel while it is still in a molten liquid stage, and that will solidify these little wells that we can load our DNA samples into before the gel actually solidifies. So that way we have somewhere to put our DNA samples. So a buffer, usually that's going to be TAE buffer, which stands for tris, acetate, and EDTA, or we can use sodium borate. I've actually used sodium borate myself in the past, but I don't really have a preference myself. So regardless, a buffer is laid over the gel, so you can kind of see this apparatus here where we have our tray with our gel in it, and then a buffer that's kind of overlaid on top of it. And the DNA samples will be loaded into those wells. So in this top-down view that you see over here on the right, you can see those wells there, and we are just going to use a micropipette to inject our DNA samples into those wells. And the DNA samples should be weighed down with some glycerol, so they should just kind of sink down to the bottom of those wells. So the whole rig is going to be connected to a power supply, which you can see in both pictures here, both the side view and the top-down view, and that is going to run an electrical current through both the buffer and the gel. So the apparatus here connects the leads to both a positive and negative electrode. So in the side view here, in both the top-down view, the DNA samples are on the left side near the negative electrode, and then the positive electrode, which is called the anode, is on the far right side. So because the DNA molecules have a negative charge, they will want to migrate in the direction of the positive electrode. So in both the top-down and the side view, from left to right, left and right, the DNA samples are going to want to migrate from the left to the right. So from the left to the right towards the positive electrode. From the left towards the right towards the positive electrode. So if you load the gel in incorrectly, if we flipped this around 180 degrees, the DNA molecules would not migrate the correct direction and they'd come out the wrong end of the gel. So that's not what we want there, and you have to be careful about things like that. Okay, so if the sample in the well contains DNA molecules of varying sizes, they are going to separate from each other in the gel, with the smallest molecules moving to the red electrode the fastest. So that's just kind of the way it goes. Something that is smaller is going to migrate much faster than something that is very bulky and very big. So if you're a football fan at all, if you took all the members of a football team and lined them up in a row and asked them to run a race for 100 yards, it's going to be probably the smallest guys that are going to be the quickest to the finish line, while the big offensive and defensive linemen are probably going to be the last. So the same principle applies with DNA molecules here. So you can look over here on the left where I'm pointing with my mouse here. That is the well that we loaded the DNA into, and then after we run the current through the gel for maybe between half an hour to an hour, you can see the DNA molecules have migrated out so that the largest molecules have made the least progress, and the smallest molecules have made the most progress in moving towards the red positive electrode there. So Agarose Gel Electrophoresis is absolutely perfect for separating nucleic acids, which are always going to be negatively charged. So this is perfect for that. Technically, you can use this method for separating out molecules of any particular variety. You could do proteins, you could do carbohydrates, you could do lipids. But here's the problem with those. Proteins are not always going to be negatively charged. Proteins can either be positively charged, negatively charged, or have no electrical charge depending on the amino acid makeup of that particular protein. So maybe some proteins this would work for, but most proteins it probably wouldn't. So for studying proteins, we are going to end up using a different method called sodium dodecyl sulfate polyacrylamide gel electrophoresis. So that's kind of a mouthful, so you can just call it SDS-PAGE. And that's something that we're going to talk about in Chapter 5, and you'll see that when we get there, we are going to treat those proteins with a chemical that actually causes all of them to be negatively charged like proteins are. So that kind of takes away that problem there. But carbohydrates and lipids, most of them do not have electrical charges, so we can't really use electrophoresis for them because they would not move to either electrode, either the positive or the negative. So agarose gels are going to be really good for separating DNA molecules that fall somewhere between 500 base pairs and roughly 25,000 base pairs. Any less or more than that range, and you're going to end up with some difficulties. Anything less than 500 is just going to skate through the gel before you have a chance to blink, and anything much greater than 25,000 is going to make very little progress at all, if it ever even gets into the gel. So depending on kind of what size of DNA molecules that we're dealing with, we're going to want to change the concentration of agarose in terms of percentage that we end up making our gel out of. So in this picture here, you can see 3 different gels that use a different concentration of agarose in making the molten solution. So on the left, we have 0.8% agarose, which would be, let's see here, that would be 0.4 grams of agarose per 50 milliliters of solution. So that would be how you make a 0.8% agarose solution. Or if you wanted to dial it up to 3% agarose, that would be 6 grams of agarose per 50 milliliters of solution. So the difference is that as the agarose becomes more concentrated, the threads of solidified agarose become more of an impediment to migrating DNA molecules here. So the smaller the percentage of the gel that is made up of agarose, the DNA is going to be less encumbered. There's going to be less impediment for it to encounter as it migrates through the gel. And then, like I said, the higher the percentage, the more impediment that DNA molecules are going to have to kind of work their way around. So for that reason, low percentage gels are going to be good for separating very large molecules. So a 0.8% agarose gel would be good for dealing with very big DNA molecules because big DNA molecules already are going to move fairly slowly, so having less impediment there allows the relatively smaller molecules to kind of jump ahead of the much bigger ones. But if your DNA sample is a mixture of very small molecules, you'll want to use a higher percentage agarose solution because there's more impediment and it will give the extra small molecules a bigger advantage to separate from the slightly bigger molecules. So just to give you an idea of how important it is to pick a good percentage for your agarose gel, if you were to try to run a very small piece of DNA on a low percentage gel, that DNA molecule may move so quickly through the gel it may kind of fall out the bottom of the gel before you ever get the chance to visualize it and see what happens. So definitely something to consider there. So ignore the title of this slide. That's a title from a previous slide, so I guess I never got around to fixing that title, so don't worry about the title there. So here's the deal. Even after you have run your gel, you're not going to see anything. DNA is colorless, so even after you separate your molecules, you can't see the DNA. So all you're going to see is just the gel. So how are you going to see the DNA? So in order to visualize the DNA molecules, you have to apply a dye that will bind to the DNA molecules and cause them to fluoresce. So the most common dye is called ethidium bromide. So this is what is called an intercalating reagent. So if you look at this DNA over here, you can actually see the chemical structure of ethidium bromide here. Ethidium bromide will wedge itself in between the stacked base pairs of the double helix of DNA. So that's what's called intercalation, when something wedges itself in between those base pairs. So it actually becomes a part of the DNA molecules, and it does so proportionally so that when you shine UV radiation on your gel, you should be able to see the DNA molecules light up. So you can actually see a gel being placed on this box that emits UV radiation, and you can actually see the bands of DNA as they have migrated through the gel, where each band represents a collection of DNA of a particular size. Now, ethidium bromide is not the only dye that you can use. In fact, a lot of people don't like using it because it is fairly toxic. It is a known mutagen, meaning that if you're exposed to especially high concentrations of it, it can mutate your DNA and possibly cause cancer. So a lot of people like using safer reagents like cyber green and things of that nature. So we'll probably be using something similar like cyber green. Now, in terms of getting the dye into your gel, there are a couple of options. You can either add the ethidium bromide or equivalent dye to the molten agarose before it ever solidifies so that the whole gel is impregnated with it, or if you don't want to do that, or if you forgot to add the ethidium bromide, after the gel has already been cast, you can actually cast and run, you can actually soak the gel in a low percentage solution of the dye and let those dye molecules work their way into the gel and bind to the DNA molecules. Either way works. I prefer to add the dye directly to the molten agarose because it's easy and there's not a time-consuming step involved. Okay, so once a gel has been run and visualized, let's talk about interpreting the results. So look at lane 1 here. So you can see we have a gel that has 10 wells in it, so we probably loaded samples into most of these wells and then run the DNA out. So look at lane 1 first. So a lot of larger biotech companies sell what are called DNA ladders, which are basically just collections of some type of DNA that has been cut with different restriction enzymes. So this is genomic DNA from the lambda phage virus. It has been cut with the restriction enzyme called HND3. So restriction enzymes are like little pairs of scissors that cut DNA at predictable places. So because those cuts are predictable, the phage DNA was cut into fragments of predictable base pair size. So we can load a DNA ladder into the first lane of a gel, and because we always know how those bands are going to migrate, we can use it as a reference or a standard to compare the sizes of DNA molecules that we are interested in. So for example, if you were to look at lane 2, it only contains one particular type of DNA molecule, and you'll notice that it's migrating somewhere in between the 6557 and the 9416 bands in the ladder DNA. So just eyeballing it, it looks like the DNA molecule in lane 2 is about maybe 7,000 base pairs, although that's an approximation. It's going to be somewhere between 6,500 and 9,500, that much we're sure of, but it looks like it's maybe about 7,000 base pairs. So you can definitely see the value of adding a DNA ladder because it's a good reference for the size of other DNA molecules in other lanes. Okay, look at lane 3. So lane 3 appears to contain 3 different pieces of DNA of different sizes. They appear to be, the bottom one down here looks like it's maybe about, 1,600, 1,700 base pairs. The one in the middle looks like it's maybe about, 25, 26, 2,700, and then this one on top looks like it's maybe in the neighborhood of 3,000, 3,200, something like that. So again, these are kind of approximations, right? So because there are 3 different pieces of DNA here, we can speculate as to where those DNA molecules came from. So this might be the result of a restriction digest, which you are going to get some experience with in your next lab session. You're actually going to cut lambda-phage DNA with restriction enzymes, and you'll be able to kind of see how the fragments of DNA migrate through an agarose gel. So a larger piece of DNA might have been cut with restriction enzymes producing these 3 pieces that you see right here. So lane 4 clearly contains a DNA molecule, one type of DNA molecule that is very, very large. So this looks probably like genomic DNA from bacteria. So it's clearly bigger than 23,130 base pairs, so it looks like it might be in the neighborhood of maybe 50,000, 100,000 base pairs. It's a little hard to tell there. So you can definitely see it didn't make much progress through the gel. It's really not that far away from the well in which it was loaded. Lane 5, you can see down here, has migrated very quickly, and it appears to be a little bit faded and a little bit streaked there. So that kind of gives you an idea of how RNA molecules would run through a gel. But gel electrophoresis is not that common in dealing with RNA molecules. It is much more common in dealing with DNA molecules. You can definitely see a lot of smearing in lanes 6 and 7. And what this usually indicates is that you have got a sample that contains thousands of different DNA molecules of all varying sizes. So you're not seeing any discrete bands, which would indicate that you've got a lot of one particular size of DNA molecule. We've got a huge collection of DNA molecules that all have different sizes. So instead of having discrete bands, it just kind of streaks down from the largest band to the smallest band, with all the bands in between making it look like that streak there. Lanes 8 and 10 do not appear to have any DNA in them, so we either left those wells blank with nothing added, or we added a sample that did not have any DNA or any RNA in it. And then lane 9, you will notice the DNA is all still at the level of the well. So the only interpretation we can really make here is that we must have loaded a DNA sample that contains DNA molecules so very large that they could not thread themselves into the agarose gel. Therefore, they were never able to migrate through the gel because they never got into the gel in the first place. So this would probably be what it would look like if you tried to load a whole eukaryotic chromosome in there, something that is millions, if not billions, of base pairs big. All right, that is going to do it for this chapter. So we have gotten a little bit of a head start on studying DNA. We are by no means done studying DNA, but we have gotten an excellent foundation to work with for the time being. And once we start talking about DNA biotechnologies again, I really think, going to be ready to dive in headfirst and talk about these sorts of techniques and details in much greater fashion. So I thank you for your attention in this chapter, and I hope you will join us next time, and we will start tackling the fundamentals of studying proteins.